that metabolomics can help to refine this follow-up. Moreover, it's currently known that there are complex interactions between tumor and its environment, in which one of the key players is host immune system. Having an approach to non-invasive imaging this would be a useful tool for therapy response follow-up. For properly studying that, we need to use preclinical immunocompetent models. So I will briefly explain some of our results which conducted us to our working hypothesis. In order to investigate response, in addition to preclinical immunocompetent models, which in our case is the GL261, we need therapeutic approaches which are able to produce either transient or sustained response. In this case, temozolomide, which is administered in an immune-friendly schedule every 6 days, which we have described in this reference. We have described the MRSI study of control and temozolomide-treated responding tumors. In this case, each MRSI spectral vector is used as a single input. So those spectra have differences and can be categorized between normal, non-affected tumors, actively proliferating or controlled tumors, and treated responding tumors, each one assigned a color. After that, with this information, we can generate the nosological image in which each spectra is assigned the color of its category. This is the nosological image. Then there are metabolomic differences between control and responding tumors that are consistently detected. But we all know that glioblastomas are very heterogeneous tumors, so rather than studying only one slice, we moved to multi-slice acquisition, generating several nosological images the same way I described before, and we were also able to calculate what we call the tumor responding index, which is the amount of pixels showing a pattern related to responding tumors over the whole tumor pixels. And just keep this tumor responding index concept because it will be important in the next slide. Go to longitudinal follow-up. Here we can see an example of a temozolomide-treated mouse. In black, we can see a tumor volume calculated from MRI with transient growth arrest, partial response, and after that a relapse. And in green, we can see the TRI, which is calculated as I just showed in the previous slide. And as opposed to the tumor volume, the TRI oscillates, going up and down with a frequency of 6 or 7 days. And this frequency matches the length of the cancer immune cycle, which would work as more or less stated here, starting with a therapeutic at day 0, which would produce cell damage and release or exposure of immunogenic signals, which would attract host immune system, antigen fragment presentations followed by T cell priming and proliferation in lymph nodes, and after that those primed T cells would just be trafficked within tumor and kill tumor cells, probably with the help of macrophages. Then our hypothesis is that our MRSI-based biomarker is sampling changes in tumor microenvironment which are related to response to therapy. To corroborate that, we have generated a cohort of control and temozolomide-treated tumor-bearing mice. And we have collected those tumor samples guided by the appearance of our MRSI-based non-invasive biomarker. And we have analyzed these samples by qPCR for different genes, for which I will discuss the corresponding results in the next slides. Regarding macrophage populations, we have found increased population in tumors treated and responding to therapy, which is in line with literature in this regard. We have already found this in IBA1 immunohistochemistry. Here we can see a chosen field from a slide in which IBA1 positive cells are surrounding a giant multinucleated tumor cell. More than that, we have observed a switch in the macrophage phenotypes towards the more M1, which M1 have pro-inflammatory and anti-tumoral actions as opposed to M2, which are pro-tumor. So not only macrophages have increased, they have switched their phenotypes. Regarding PD-L1, this was also found increased in tumors treated and responding to therapy. PD-L1 is the programmed cell death ligand 1. This is a well-known axis pathway for tumor-evading immune system attack. Then, cells displaying high levels of PD-L1 can interact with T cells and protect themselves from the attack. So it's sensible to think that in front of sustained host immune system attack, so cells try to escape this attack by overexpressing this ligand. Well, in this case, rather than showing individual values, we concentrated on whether those values would be correlated between them and how this correlation would change under therapy. This is still an ongoing work, and it's not straightforward to interpret due to the complex role of metalloproteases in cancer issues. But we will just highlight some correlations which appeared after treatment. For example, this correlation between ADAM17 and MMP14, which was not present in controls, but was highly positive after treatment. ADAM17 was described to be correlated with more presence of M1 antitumoral phenotype of macrophages, which is in line of our results. And MMP14 was described to increase after some therapeutic approaches. The other correlation which appeared after treatment and was negative before and became positive after treatment was between ADAM8 and PD-L1. And both of these factors are related to tumor escape to therapy and may be reflecting the adaptation of cells after treatment, trying to escape the next round of treatment. Our conclusion is that our biomarker may provide information about host immune system against tumor, which is not currently possible in clinical follow-up protocols, and this may have a translational potential. And that's all for today, and thank you very much for your attention.